Oh my gosh. I really don't know. I'm trying to roll my page. Yeah. Oh, okay, that's all it is. Water. So we're gonna begin getting scrolling with our. As you will shortly begin to realize, if you have not realized it yet, we've got a lot to get through this year. So is everybody able to find that folder of your copy of the lecture slides? Yes. Okay. Excellent. So we are going to begin with the one numbered 1. So today we're going to be talking about how we simplify the body plan in some of our diagrams, and we're going to be talking about homeostasis. Okay. We are also recording on Zoom, so you guys will be able to see this in your YouTube playlist later if you miss anything. So we are going to start out by recapping a little bit of what trying to get us to do in that activity at the end of last time. Part of the point was just to get us standing up and our blood moving a little bit on the first day of class, because I know everybody's pretty bleary. And also to make you realize that you need to look up your lab section. But there was a little bit of content in there. We're going to go over it formally right now. So our first objective is to think about water in the body, mostly made up of water, and think about where that water is located, and think about the idea that that water is located within compartments. As we move through this course, we're also going to be talking about things moving between different compartments, different fluid environments. Talk about that a ton when we get to the kidney next semester, which is all about fluid balance. But we'll be talking about it very soon when we start to talk even just about cells. When we talk about neurons and how the brain works, we're going to be talking about how do things move from the internal environment of the cell to the external environment and vice versa. So understanding that water is everywhere and how we talk about those different pockets of water is a helpful place to start. We're also going to talk about homeostasis, which is hopefully a word you've heard before in your prerequisite classes or in high school. But we're going to think about that and its significance to the human body and how to live our lives. As we're talking about homeostasis, we're going to define some terms, and a super important one is negative feedback. Also talk about positive feedback, but negative feedback is a really important concept to master as we're moving through this unit. Okay, so this is a figure from your textbook. So if you were following my helpful tips on how to kind of review before class, you might have noticed this picture is in your slides and in your textbook. So it's a thing to kind of look in and get a feel of. Okay. So this is meant to be a simplified body plan. Now, I'm a human anatomist, so I kind of hate this picture because it does not look like a human body to me. But when we're thinking about physiology, that's actually helpful. Okay. So it's simplified and all square and gross to show you the relationships between general types of structures in the body. Okay, so it's missing lots of parts because we can generalize. We have lots of cell types that aren't shown because we can generalize. We're just trying to look at relationships here. The first thing we're looking at in this diagram is the difference between what's outside your body and what's inside your body. So we are separating the external environment of you, yourself, or human body. Right? The external environment, we're thinking about like the air, you're swimming, could also be water. That external environment, separating out from the internal environment of the organism that we are talking about. us. These are separated by a specific type of cell layer. So this is separated with epithelium. So your epithelial cell layer, this would be your skin, but you're also going to see epithelium lining other parts of the body as well to create this really tight seal so that we can control what goes in and out. That's our first piece, external environment versus internal environment. I want you to take a look for a second, and if you didn't look there before class, I want you to just get a feel for it. So go ahead and look for where you can see the lungs, where you can see the digestive tract, see if you can find the kidneys, and see if you can find the heart. Take a second to kind of process what's going on. I'm going to go ahead and start pointing those out. Right. So lungs, we can see here, right, this baggy structure. One of the reasons we're looking for these organs specifically, and why these are the organs that are actually listed on this diagram, there are any other organs that you have that aren't pictured, is that these ones are particularly important ones when we're thinking about understanding these different fluid compartments, essentially. So one thing we would notice about the lungs here, right, is that notably, you see this purple color inside, right? So the external environment, it's important to realize when we're thinking about physiology, isn't necessarily what you would think about if you're just talking to a friend in normal human life. Right, so for us in physiology, the external environment is anything that's touching the external environment, essentially. So the air inside your lungs, the tasty oxygen and carbon dioxide, right? It's still in communication with the air touching your lips, touching chalkboard. So it's still considered the external environment. We haven't pulled it into the body yet. It's not touching all your different cell layers, all your internal systems. Same thing is true when we're talking about the digestive tract, right? So if you eat a taco and that taco is going through your digestive tract, I just realized I don't know how to draw a taco, sorry guys. Right, you are not part taco at that moment, that taco going through your digestive tract, it's still, for our physiology purposes, part of the external environment. And none of that taco is going to be part of your internal environment until we start to absorb specific nutrients, specific biomolecules, which we'll get to biomolecules in this unit. We'll get to the digestive system next semester. Okay? So still in the external environment, but we do have pathways that allow things to move from external to internal. But that's going to be a very controlled process. When we eventually do get to our GI unit, we'll be talking about all sorts of receptors and enzymes and cell types and how things travel across that layer, right? We have regulation, we have control, we have to do something to transfer things between compartments. Kidneys are also partially communicating with the external environment. So urine comes out of your body through excretion, and those tubes actually start way up in the kidneys. This is why it turns out you can get like a kidney infection from a urinary tract infection, or someone on TikTok who said they got it from just like holding their pee too long, right? Because we back up all the way up to the kidneys, and we actually do have this communication open with the external environment. Without a kind of control going in. The reason the heart is on here is something different. So we have the heart here. The heart is our muscle pump. So the heart is part of the internal environment. It's fully inside the body. It's not touching the air outside, okay? But it is touching a separate compartment that we're looking at, the cardiovascular system. So cardio is our root word for heart. Vasculature refers to like arteries and veins, so blood vessels. That's what vasculature is. So cardiovascular, the heart and blood vessels. So that forms a independent sort of pathway that we'll think about as we work through this unit. So system involving your blood also is going to be regulating what comes into it, what goes out of it. Super important that stuff can go in and out because it's traveling all the way around your body, distributing gases, pulling in nutrients, doing all sorts of important stuff. But we don't want just anything getting into your bloodstream, and we want to control what leaves your bloodstream at specific times as well. Okay, so when we move forward, we'll talk about the process of filtration, moving fluid and other things out of your bloodstream, talk about reabsorption. Sometimes we move stuff back after we filter it. Sometimes we secrete things, so we can take things all the way from your bloodstream out to the external environment. And we looked at absorption. So if you're eating that taco, the way you distribute all those biomolecules around your body is that you absorb them all the way into your bloodstream. Your blood travels somewhere else, feeds another organ, say, I don't know, your knee, something. Okay. So that's how we're looking at this picture. So we're differentiating the external environment from the internal environment by that epithelial membrane outside. Okay, so we had square, give us our digestive tract here. Right, body was continuing on the other side. I'm just redrawing that picture, but worse. So I just like to kind of talk through things as we're doing them. Okay, and we had the lungs here, also lined with that epithelium. So air in the lungs, food in the stomach, air in the bladder, anything that's touching that external environment is external to the body barrier. Our internal environment was anything contained in here. Okay. Now your internal environment, anything inside your body, we're going to think about that as like the immediate environment of most cells. What we mean there is like, if you have a cell that we're thinking about, I don't know, like a fat cell right here in your belly, right? We're going to want to be able to think about what environment is that cell existing in? What are the concentrations of different chemicals outside it, inside it? So a random cell of your choice, choose one, is going to have an environment around it that is external to the cell, but still internal in terms of your body, okay? And we're going to be thinking about that environment as we think about what passes in and out of cells within the body. Okay? So when we think about that internal environment, if we're just surrounding cells but not inside them, it's going to be considered extracellular fluid, extra for outside, cellular for the cell, so outside the cell. And we do specify two different types. So we're going to specify whether we're out somewhere in general types of tissue, say your muscle, wherever. So there's going to be fluid surrounding the cells in the tissue of your different organ systems, your different regions of the body. And so tissue fluid surrounding random tissue, we refer to as interstitial fluid. And so between the tissues, I think is probably the breakdown of that word. And so it's going to be around just our generalized cells. So in the previous diagram, those cells would have been that random clump that was just like that honeycomb shape, right? Those are just representing your generalized kind of tissue cell. So those would be surrounded by interstitial fluid. But we specify that the cardiovascular system is different. So we had our heart, and we had the blood going around separately. Okay. You think about blood on its own. And so this cardiovascular system has fluid in it, also has a different kind of distribution of cells. I'm going to draw the blood cells swimming around in black here, right? So these blood cells are still in a fluid environment that is still outside the cell, and that's the plasma. So that would also be a type of extracellular fluid. It's not inside a cell. It's not surrounded by a cell membrane. Okay. But it's got its own name because it's within our cardiovascular system, and it's going to have different properties, different kind of factors that we think about. When we talk about these different compartments, we're going to be thinking about like different pH, different concentrations of things. So the blood is kind of doing something. So we think about it separately. So we have exchange between these different parts of the body, these different compartments. So we have exchange from the external environment to the plasma, so all the way into the blood. We have exchange between the plasma and the tissue fluid. We have exchange between the tissue fluid and the cell. And we'll have specific terms that we use to describe this. So they're on the diagram. I'm going to tell you, these are not going to be super important for this specific unit. They are worth knowing and preparing because we will talk about them as we move forward, especially when we talk about the cardiovascular system, when we talk about the GI system and the renal system, but we'll go through them again at that point. What we want to really emphasize right now is that stuff is able to move between these different parts of the body. Okay? So we'll have specific terms that are based on the direction and are based on where we're coming from, where we're going to. So these are some summaries of the locations that things are moving, the mechanisms they're moving by. And this is kind of what I was talking through as we were looking just at the diagram, trying to find some things. So this is just noting that for the external environment, right, we have exchange of gases with the blood. Pick a different color. Okay. So we have gases being exchanged with the blood. So we'll talk about that extensively when we talk about the respiratory system. Okay. We have different molecules being exchanged between the GI system and the blood. Okay. So we have, say, some carbs, some sugars moving into the blood. We are secreting out all sort of waste products into the GI tract. Same thing going on with the kidneys, right? Especially that removal of waste products through filtration and secretion. They're going to be really important when we get to our renal unit. And this is just kind of demonstrating where these different processes are going. But consider this kind of overview and big picture stuff for now. We'll get into the nitty gritty in those units specifically. We do want to, for now, understand the difference between internal environment, external environment, and those different fluid compartments where we find that fluid. So go ahead and answer to yourselves, which of these things would be considered part of the body's internal environment? When you've looked at it yourself, go ahead and whisper to a neighbor to double check. Would someone like to volunteer to tell me the answer? Oh, said they would volunteer. Hyper here? Yeah. You want to tell me? We're gonna go with D. Yeah, correct. All right. See, not so scary. Thank you for volunteering. everyone's hearing. I really appreciate it. Okay, cool. Yeah, so the extracellular fluid is still part of the body's internal environment because it's surrounding your cells, but it's not out in the, right? It's not outside the epithelia. It's just outside the cell. It's still inside your body. But answers A through C are still in communication with the air, right? There's like a hole, right, that touches the external environment of the body, which is why A through C don't work for us. Okay. So looking at these fluid compartments, we divide it into compartments. Each compartment contains fluid. The compartments are separated by epithelial membranes. So epithelium is a specialized kind of cell type of cell structure that forms like really good tight seals. We have something called tight junctions linking those cells together. They form a nice like Ziploc baggie essentially around whatever they're surrounding. So we have epithelium as part of your skin, right? Your epithelium is your skin separating you from the external environment. But we'll also have epithelium lining the lungs. We'll have an epithelium membrane lining our bloodstream as well, right? So this is separating out different compartments. So epithelium, however, can be crossed under certain conditions when the cells are in a certain state, depending on how your body is regulating things. And so can the membranes of cells for that matter, right? So we have semi-permeable cell membranes. Whenever we see that word semi-permeable, permeable means something can cross. And semi means not everything can cross, right? This is selective, essentially. And in this unit, we will be talking about transport. So at some point soon, you want to go review active, passive transport. That'll be kind of the end of unit 1. So getting a grip and kind of refreshing your brains on that would be a good idea. Okay. So transport is going to occur between compartments and also across our cell membranes. So when we're talking about all the water, we'll talk about total body water. That's just everything all together. Okay, so total body water, it's the water and anything dissolved in it. They dissolved in it. Often we're thinking ions. So go ahead and remind yourselves over the weekend if you're fuzzy on ions. Ions are really key to understanding what we do in the second half of this unit, which is what most people kind of struggle with when we get there. So remember, ions are things like sodium ions, chloride ions, you have anions and cations, but really what all they are like atoms that have a charge on them. We're going to be thinking about that charge. We're going to be thinking about what that charge does, what it means when we think about things moving between our different types of fluid, between our different fluid compartments. So probably won't be next week, but this is a heads up for you for things to kind of flip through your notes on before we get there. Okay. So it's going to include in the total body water, the solution within the cells, our intracellular fluid, as well as the solution surrounding cells, so the extracellular fluid. Okay, so if we were drawing a cell, which I will draw you endless, probably not very good cells, right? So we're going to have, consistent with the figures in your book, they usually use yellow for the outside of a cell. Okay, so if we have a cell, we're going to have fluid inside. Right, we're going to have fluid inside, that's our intracellular fluid. Upside down water drop. Intracellular fluid. This is in the next couple slides written out in words. Okay. And we also have water outside, solutions outside the cell, your extracellular fluid. Okay. So when we talk about transport and things that drive transport, you're going to be thinking about how and why. So we have something, say, in the extracellular fluid. Under what conditions would it move in to the cell? Under what conditions would it move out of the cell? Because usually our ions and other dissolved materials are going to be distributed both inside and outside a cell, but just in different concentrations. That's a preview of where we're eventually going. So this is just that picture again. Our body fluids are subdivisions of total body water. TBW is just all the water in the internal environment, not specifying where. Compartments refer to just a location that's surrounded by a membrane, so separating where we would see control across that membrane. Okay. And we don't really distinguish between the name for the body fluid or its location. So the intracellular fluid, we're just talking inside the cell. You don't need to use a different word for that spot versus that fluid. Okay. So intracellular fluid here is in blue. So we can see the intracellular fluid inside our random tissue cells, and we can see it inside our blood cells, right? But it's inside a cell. Inside a cell doesn't include what cell. Our extracellular fluid, our ECF, is in blue here. So it's outside a cell, but still in the internal environment, so still in your body. And so we see it here next to the lungs, but we also see it here in the cardiovascular system, just next to the GI tract. So extracellular fluid is everywhere. So that's our division between ICF and ECF. And if we added those numbers together, we would have highlighted everything in blue. So it would add up to the content, the total body water, because we just colored everything in. Okay. But we do get a little more specific eventually when we talk about the ECF. Okay. So the ICF, we don't have to specify, right? So intracellular fluid, the ICF, and this is a good time for me to know kind of the structure of your slides. So your slides basically work like guided notes. Almost everything is there, but you'll occasionally see blanks. That's so that you just kind of try to keep awake, right? I know this is a lot of me talking, right? So this is kind of a scavenger hunt while I'm talking. So when things appear in red up on the screen in my slides, that's what's missing from your slide. So you can just go ahead and fill those in. Don't get too panicked about searching for things while I'm talking here in lecture if it's stressing you out, because you do have these recordings up on YouTube. So if you miss one or two by the end of the lecture, that's just a good tip for you that you weren't paying attention, or you were paying so much attention to me and my face that you forgot to write something down. Totally fine. Go back and you can find it. All right, so ICF is our intracellular fluid. So anything inside a cell, doesn't matter if it's in a blood cell, a tissue cell, an epithelial cell, it's all intracellular fluid. So basically the fluid in the cytoplasm of the cell. Okay. But extracellular fluid, we get a little more picky because blood is different. Okay. So extracellular fluid, if we just talk about it generally, it's anything outside a cell, right? So extra outside versus intra is inside. Cellular, a cell, okay? But under certain conditions, we care whether we're talking about the bloodstream or just around tissue, especially when we get to the point of talking about, okay, how does stuff get into the bloodstream? How does stuff get out of the bloodstream, right? You're gonna have some communication between this interstitial fluid and the plasma, which is why it's important to have different words for them, okay? So plasma, if you donate plasma, right, we're talking about the fluid that is surrounding the blood cells specifically. So that's this first image here, right? So in blue, we're showing that water, okay? So that's the plasma inside your cardiovascular system, but outside the blood cells, which is why it still counts as extracellular. Interstitial fluid, not inside the cardiovascular system because we already have a word for that. So it's just all the fluid between tissue everywhere else. So everything colored in blue on this next image. Any questions about these divisions? So we could add them all together, and this is the slide that I very quickly showed you at the end of Wednesday. So this just shows you that it's true. Your total body water is composed of your intracellular fluid and your extracellular fluid. So everything inside a cell plus everything outside a cell, those add together. And then separately, we can divide further. That extracellular fluid is composed of your plasma plus your interstitial fluid. Those add together and tells you everything that's outside a cell. Okay. So up to here would be just to make sure that you study and practice ICF versus ISF. I think the concept is hopefully pretty clear, but sometimes the fact that those abbreviations look similar can trip people up. But they are different in important ways, so it's important to realize that. Okay, so in terms of your body, we use the physics person. So when we give you amounts in this class, when you're in the anatomy lab, you'll see there's actually a lot of human variation, just like tons and tons and tons. Things will not always look the way you expect them to. They won't always look the way I expect them to as an anatomist. So when we present amounts, this is usually based on the same person that I think they used at least for all my high school physics books, which is a 70-kilogram male. I think it makes their math easy, at least for some things. So in a person of that size, the total body water is about 42 liters. That's where these numbers are coming from. But overall, that'll be about 60% of body weight. Separated, 40% is going to be inside cells, 20% is outside cells. And then of that 20% of it is in the plasma. The rest of it, the 80%, is mostly meandering around between your various cells. All right. I think we covered this, but this slide should point out to you as you're going through the textbook, as you're going through different sections, these checkpoint questions will appear. And they're basically just checking that you digested and understood that section of material, that section of the book. So I'll pull them up in class sometimes, but even when I don't, they're worth going through and thinking about. They're not things that are directly stated in the text always. This isn't like a hunt and pick kind of activity. This is, if you understood what you were reading, hopefully you will be able to describe or explain or understand the answer to these questions. Okay, so they're useful for kind of testing the comprehension for yourselves. We are going to move on to the concept of homeostasis. Okay. So hopefully you've heard this word before, but kind of fun to step back and think about the fact that your body manages to stay a consistent temperature, consistent blood pH. You manage to have the same amount of blood volume practically your whole life, right? There are certain things about your body that don't change. There are certain things that are predictable, which is part of why we can tell when something goes wrong, right? If one of these regulated variables that we're going to talk about is out of whack, right? We know that something's happened, right? So it's helpful to understand the process of how we regulate these things. It's important, especially when we think about disease and things going wrong. Okay. All right. So we're going to be thinking about the concept of homeostasis, right? So stasis, I think, actually refers to a place in Greek, but homeo refers to same. I don't know. Regardless, we're really just talking about the fact that the internal environment of your body is regulated to keep some things consistent, keep them regular. It's really important that your body can maintain a constant internal environment. Okay. I love AC, but we can't do that much about the temperature outside, right? We can't really consistently regulate our external environment. But it's really important that we can regulate the temperature inside our body, right? So when we talk about homeostasis, we're talking about maintaining a consistent internal environment for the body. If you turn a light on and off, you turn the AC on and off, that's not homeostasis. That refers to your external environment. Homeostasis is the physiological processes that keep our temperature internally. We'll talk about different things in the internal environment that are regulated. The easiest one to think about at first from your lived lives, I think, is temperature. But we'll also see that the volume of things, especially your blood and your fluids, also a thing that we maintain. Right? Composition of different levels of different chemicals in different pockets of fluid in your body is also regulated. So we'll talk about those as we progress through, right? As we progress through the different organ systems. So regulating our different regulated variables is going to require that our different organs work together. We can't do it with just one organ. There's not one organ completely in charge of everything. You're trying to maintain homeostasis, and we have to collaborate. Okay? If we disrupt homeostasis, the regulated variables that we talk about, the things that your body really, really tries to control, are important enough that if they get disrupted for too long or by too much, it can result in death. Right? If your blood pH goes out of whack, that can be very quickly fatal, right? If your blood volume goes down too far, also fatal, right? You can bleed out, right? So these things are important to keep consistent. They're important in order to maintain body function. Okay. This is usually a process that's controlled by negative feedback loops. Diagrams of negative feedback loops can sometimes make it harder than it is. All we're talking about when we talk about a negative feedback loop is that we see a change, right? Something happens, right? Your temperature goes up, whatever. But you can think about it as like an interpersonal relationship too, right? Like you change, you say something to someone, you get negative feedback, and then you go back to what you were doing before, right? That is really all we're talking about when we talk about negative feedback loops, is that we see a change in one direction and then a change back to the original state. A positive feedback loop would be if we see a change and then someone goes, okay, great, that's awesome. And then you keep going like a big snowball effect. So that's really the only difference between negative feedback and positive feedback. We are going to look at science graphs related to that, but the basic idea is pretty similar. So when we're talking about homeostasis, we're going to have x-axis, oops, sorry, that's a y-axis and an x-axis. Okay, so usually we're going to be looking at something we're measuring over here on the y measurement. And we're going to be looking at time on the x-axis when we're talking about homeostasis specifically. All right, so we're going to see that we measure, say a temperature, say it jumps up, right? So jumps up, you take some Tylenol, your temperature goes back down, right? That wouldn't be your body regulating, that would be you regulating, but your internal systems work the same, right? So your measurements here might be hour 0, hour 1, hour 2, hour 3, so on. Okay. So this is the basic structure you're going to see for these homeostasis diagrams. We want an everyday example of this. I am not so awesome as a car person, but I'm going to try to relate this for you. Okay. We could do the same kind of experiment with like how you drive up a hill. right? So we have something we're measuring over here on the Y. So we're going to be thinking about trying to maintain a constant speed as you go. We've got some really steep hills here in Duluth, right? If you want to just speed up them at the same rate as when you're going on flat ground, you're going to have to do some work, okay? So speed over here. And then on the x-axis, we have our time, okay? What's going to happen? We're cruising along at 60 miles an hour, okay? This is going to be our regulated variable in this scenario. As you start to go up the hill, gravity is pulling you down, right? You got things holding you back. So we start to see a dip in our speed, okay? Now, when we see a dip, we're looking at a graph like this. We're going to formally, scientifically refer to this as like an error signal, right? Error because we want to keep things at 60, right? Now we have a difference. So that's an error signal. It's going to signal to your body, or in this case to you, the driver, that you're going to need to have to change something if you want to take yourself back to that set speed, that set temperature, that set blood volume. So we see an error signal, which is going to trigger mechanisms to bring you back to where you started. So bring you back to that set point. And in this case, right, you're going to hit the gas pedal, right? So you notice your car slowed down as you started going up the hill. If you want to speed back up, you have to hit the gas. And you speed back up, that error signal gets smaller, smaller, smaller until it's gone and you're back to the set. So your body works the same way as this, except instead of you and the gas pedal, it's your brain and different physiological mechanisms. So the thing that we're measuring on that y-axis, we're going to term our regulated variable. Okay, so the regulated variable is the thing that we're maintaining, and therefore also the thing that we're measuring in this graph, right? So if we're interested in something that's maintained, we better measure it, otherwise how would we know it's changing? Okay, so that's our regulated variable. So temperature, we mentioned, so blood glucose concentration is another thing that's a regulated variable. So the sugar in your blood, essentially. You have or know anyone who has diabetes, you've probably thought about this. Your textbook likes to use diabetes as an example for a lot of things, right? But having the right blood sugar is really important to functioning normally, right? You have really low blood sugar, really high blood sugar, it causes problems. It causes problems for your organs sometimes, or just causes weird cognitive effects, right? So it's something that your body wants to regulate. Blood pH, also regulated. Levels of sodium are regulated. We're going to see sodium over and over and over and over again throughout this course. So worth knowing that ion specifically, thinking about it. So sodium, reminder, ion, Na+. Really, really, really vital for the function of our body. Okay. So that's our regulated variable that we're thinking about. Our set point is what we are trying to maintain in terms of like our number, our measurement. So for this car speed, it was 60. For your temperature in your body, right, 98.6. That's our set point. Okay. I guess we should talk about it in Celsius. Celsius, 37 degrees Celsius. We are going to be using metric in this course, because science uses metric. Okay. Blood glucose, our set point is 100 milligrams per deciliter. Blood pH should be 7.4. Those measurements, their values is the set point. The thing that we're measuring is the regulated variable. Questions so far about homeostasis and this kind of idea? Like starting with that are understandable. Okay. So more turns. We have already our thing that we're measuring. So here, our example, our regulated variable is blood glucose. Our set point for normal blood sugar level is 100 milligrams per deciliter. And we know that we're looking at this over time because we're looking at maintaining that blood glucose level consistent over the whole day. Okay. So we start out with a blood glucose of 100. We may see after we eat something really sugary, right, that our blood glucose level rises, right? So it goes up high. Okay. This produces an error signal. So the error signal is just the difference between that new higher measurement and our set point. Error, we're not at the set point. Okay. And then we want to decrease that error signal until we're back to the set point. Okay. So once we activate our regulatory mechanism, whatever that happens to be, we're going to decrease that error signal. And in this example, we're also decreasing blood glucose until we return to normal. So that's a process of negative feedback because we go in one direction and then we come back to our set point. I do want to point out that if you suddenly had your blood glucose level drop, right, go down, this would still produce an error signal, right? Like this is still not at the set point. This is still not the right number. So we would still have other regulatory mechanisms that bring you back up to the set point, okay? And both of these would be examples of negative feedback. So I want to be careful that you don't think a value going up and then back down is what negative feedback means. You want to think about it as a change in one direction, and then you go back to the original state. Okay, so it doesn't matter if your blood glucose goes to 100 to 125, or if your blood glucose goes from 100 to 90. Both of those directions produce an error signal. And then we change back to the original state. We go back to our set point of 100. When we're returning to that original state, both of those, therefore, are examples of negative feedback. So negative is just telling you that we're doing the opposite of the change rather than specific numbers going up or down. The only number that would consistently go up or down is that the error signal gets smaller eventually. So the error signal is getting smaller. This error signal on the bottom would also be getting smaller, and my zigzag is getting smaller, even though the values are getting more negative for our measured regular. Questions about negative feedback specifically? Negative feedback is a super important concept in this course. So if you have questions that you don't want to ask in front of the class right now, email them to me, to my office, talk to TAs. That is a particularly important concept to build off of. I'm going to just mention what's on this slide, and this will be our last thing for today, and we'll just pick up for this on Monday. But we're going to think about general pathways that allow for this regulation, these regulatory mechanisms. Okay. So we're going to be thinking that regardless of what regulated variable we're talking about, we're going to have a stimulus. The stimulus is going to be like your regulated variable changing, right? So if something produces an error signal, your body is going to have some kind of sensor to sense that change. Usually this is going to be some kind of protein embedded in a membrane, something like that. You have different types of receptors that can sense the change. Okay. We're often going to go up to the brain to certain little areas in the brain that act as integrating receptors. So we may be getting information from multiple different locations in the body about the state of that regulated variable. We're going to want to pull all that information together. And then we're going to have effector organs. I guess I should step through right quick. So we're going to have nerves going out from the brain to the effector organs. They have the effect. They're what actually does that negative feedback in terms of like pulling your blood pressure back down, changing your glucose level back to what it should be. So we'll pick up here next time. Practice, I know we've only had one lecture. Really easy, but we're going to start making sure that we understand the format of the quizzes. So for Monday, I'll put up a quiz. Sometimes that would be done before the end of the day. It'll just be 3 questions essentially about today's content. Do it wants to check your understanding first, then take it a second time with your notes if you don't get everybody right. Bring those in by Monday. I'll take your highest score, and then we'll get in the rhythm. Right. Have a great weekend.